In eukaryotic cells, DNA takes place in the nucleus during the S phase of the cell cycle. DNA is made of two long strands of nucleotides that coil around each other to form a double helix structure. These two long strands are antiparallel and complementary to each other. One strand runs 5' to 3' while the other runs 3' to 5'. The 5' and 3' refer to the amount of carbon in the sugar bases of the nucleotides. The nucleotides are made of three parts: the phosphate group, the 5-carbon sugar, and the nitrogenous bases. The phosphate group contains a phosphate atom and 4 oxygen atoms. The 5-carbon sugar is known as deoxyribosine DNA, and the nitrogenous bases includes 2 purines, adenine and guanine, and 2 pyrimidines, thymine and cytosine. Each purine binds to a pyrimidine, forming hydrogen bonds. Between adenine and thymine, there are 2 hydrogen bonds, and between guanine and cytosine, there are 3 hydrogen bonds. These bonds are relatively weaker compared to the covalent bonds known as phosphodiester bonds between a sugar base of one nucleotide and a phosphate of another. The backbone of DNA is made of a sugar phosphate group, which provides support for the DNA strand, while the nitrogenous bases forms the rungs, which hold the two strands of DNA together. This ensures genetic information is stored accurately. The process of DNA replications results in 2 sister chromatids, as the new strand contains one parent strand and one newly synthesized strand. The purpose of DNA replication is to pass on genetic information, maintain the genetic stability of an organism's genome, and to ensure its functionality and growth.
When the strands begin to separate, it starts at the replication origin. The enzyme DNA helicase then binds to the origin and starts to unzip the 2 DNA strands by breaking the hydrogen bonds that are keeping the nitrogenous bases together. As the DNA unzips, it creates the replication fork shape that looks like a Y. The replication fork consists of 2 strands: the leading strand that runs from 5' to 3' and the lagging strand that runs from 3' to 5'. The DNA helicase moves away from the opening of the replication fork, moving towards the 3' of one strand and the 5' of the other. It continues to separate them as helicase moves in both directions. DNA replication happens at many sites along a molecule. This causes for there to be couples of replication forks, which ultimately create replication bubbles. The bubbles will increase until they join to form 2 strands of DNA. Our rate of DNA replication is 50 base pairs per second. As the strands are getting separated, another enzyme called topoisomerase or gyrase keeps the strands from getting tangled. As the strands are being separated, they are starting to coil and hold a lot of tension. Topoisomerase cuts the strands, which allows them to unwind, and then rejoins them together, which relieves tension on the backbone of DNA. There are also single.
The enzyme RNA primes add small complementary RNA segments onto the strand at the beginning of the replication fork. Primes are needed because DNA polymerase III can only add nucleotides to prime nucleotides. As an RNA prime is in place, DNA polymerase III begins adding DNA nucleates to the third end of the RNA prime as the replication fork continues to open. Enzyme RNA primes add small complementary RNA segments onto the strand at the beginning of the replication fork. Primes are needed because DNA polymerase III can only add nucleotides to prime nucleotides. The enzyme prime adds RNA nucleotides to complementary base sequences. These RNA nucleotides act as a prime for DNA nucleotides. As an RNA prime is in place, DNA polymerase III begins adding DNA nucleotides to the third end of the RNA prime. As the replication fork continues to open, the leading strand as it continuously builds by DNA polymerase III moving towards it. On the lagging strand, DNA polymerase III must move away from the replication fork, which means it's copying in chunks, what's called the Okazaki fragment. As DNA polymerase III is moving from the helicase, it must constantly return to copy new Okazaki fragment chunks and primed pieces of DNA. As the Okazaki fragments runs into another, the enzyme DNA polymerase I remove the RNA prime and replace them with DNA nucleotides. Then the enzyme DNA ligase joins the segments with phosphodiester bonds. At the end of the replication, there are no more RNA primes, only DNA nucleotides. The RNA prime are replaced with the DNA bases by DNA polymerase I, and the fragments are joined together by DNA ligase.
There are many errors that can occur during replication. The most common is base pair mismatches that distort the shape of DNA. This results in the hydrogen bonds forming improperly, causing polymerase III to be unable to move further. To solve this, polymerase reverses and replaces the base with the proper one and then moves on with replication. Next, there are more rare errors that happen as well that go uncorrected by DNA polymerase III. To attack these errors, repair complexes made from DNA polymerase I and DNA polymerase III locate the distortions and remove the mismatched bases from the new strand. The gaps are filled by DNA polymerase III, which gets the correct base, and then they're joined together by DNA ligase. Repair complexes can also be used to correct damage to DNA from chemicals or radiation. Without these repair mechanisms, DNA would accumulate many errors which could change the proteins that get made, or even lead to cancer.